So I just want to say good morning to everyone on this fine Monday morning and welcome you to the Social and Behavioral Research Branch's semi-annual, would that be right, seminar series. We come together twice a year to highlight scholars who are engaging in innovative research at the intersection of the social and behavioral sciences and genomics. So today, it's my great pleasure to introduce our speaker, Molly Bray. Dr. Bray began her academic career in kinesiology, after which she got the research bug and earned her doctorate in human and molecular genetics from the University of Texas Graduate School of Biomedical Sciences. And I think we have another alumni from from that program, Paul Liu, who's our scientific director of NHGRI. Sorry, I'm embarrassing him. But so what are you? The Deputy Scientific Director of NHGRI. Dr. Bray served as the director of the Heflin Center for Genomic Sciences, Genomics Core Laboratory at the University of Alabama, Birmingham, after which she returned home to Texas and is currently the Susan T. Jastrow Chair for Excellence in Nutritional Sciences and the chair of the Department of Nutritional Sciences at the University of Texas, Austin, and the College of Natural Sciences. Dr. Bray has authored or co-authored well over 100 publications in the fields of sports science, nutrition, dietetics, physiology, biochemistry and molecular biology, endocrinology, and metabolism, as well as genetics. Dr. Bray is an internationally recognized expert in the genetics of obesity, where her research focuses on the relationship between energy balance and lifestyle factors such as exercise, nutrition, and circadian patterns of behavior. And so I'd like to give Dr. Bray a warm welcome today, and she's going to share with us her vision of the multi-omic view of exercise response. Actually, you don't need
this is on. I think this is on.
Yes, it is.
Yeah.
I'll leave that there. It's scary.
Well, thank you so much for the invitation. And normally when I'm in Bethesda, I'm reviewing grants. So this is a nice change from that. And I will share with you today some of our genetics of exercise work. It's almost getting ready to get submitted. So, and then if we have some time, I may talk a little bit about some of the circadian rhythm stuff that we're doing. So this is not a new message for anybody. We know all that physical activity, inactivity is a significant public health problem. And many of us are kind of tackling that problem in different ways. In my lab, we want to know whether or not genes have anything to do with that, or whether obesity segregates in families, as it does in this. That was the corny geneticist joke for today. So there's a lot of evidence that physical activity has a genetic basis, but I love that the focus today is on behavioral genetics, because I can remember when I was a grad student, nobody thought you could never find a gene for behavior. It was too crazy and complex, right? And now it's like, of course we can find those genes. And of course we acknowledge that genes have a lot to do with behavior. So this is some old work from Tim Lightfoot's group that basically shows, and a lot of this old work has been replicated many times, to show that spontaneous physical activity is widely varying across strains of animals. And this is a little bit of data from the circadian rhythms study that we are working on. So you can see those little guys go insane on that. That's not speeded up. That is the real time. They like to play on the little thing. But those animals, those are FEBNs. They're actually not on Tim's graph there. And I asked him, did you ever put them on the graph? Because they would have been way past C57LJs. They're really, really crazy, spontaneously active. So that variety of distribution of activity is one evidence that there may be some genes going on. And then these are studies that again, these kinds of studies where physical activity traits have been bred into animals specifically have been going on since the 1970s. So on the left there is some data from DeFries's group that basically from the 1978 that basically showed they were breeding animals for their open field natural physical activity in an open field. And people always think the open field is that they're running around with the roses and the grass in the meadows, but no, open field, sorry for mice, is not anywhere look like a field. It looks like this, and basically you track the animals as they move across this grid. And then there's many, many groups now who have done inbreeding studies, crossbreeding studies, taking high active and low active animals. And basically what you're seeing here is this is movement throughout the grid. This one is revolutions, or this one, let's see, I should put my glasses on. Total revolutions, and basically you can breed this stably into animals across traits. What's interesting is that the genes underlying that behavior are distinct. If you breed animals for spontaneous wheel running, they aren't necessarily super active in the open field, and vice versa. And that's an interesting observation. Animals where this is just some data from Tim Garland's group, where animals were bred stably over 13 generations, and they're about 2.5 times, they run about 2.5 times as many revolutions. They run faster and slightly longer. But faster is more the reason why that they're running so much more. And just to give you a sense, if you ask this very active mouse to walk from Bethesda to the White House, it would take that animal about 2 days and 6 hours, and its wild-type counterpart would still be back chilling out at the National Zoo. Corny joke number 2. Okay, so what's been shown pretty robustly, again, this is some pretty old data, is that it's a neural trait that's being bred. So while, and actually Ted Garland was explaining that when they were breeding their animals, what they do is they give the animals access to the wheels for a day or two. So they count their revolutions and then they breed them. So they're not training them. It's not a training trait, and that's what most people think is being bred into these animals. But it's actually it's the neurally driven propensity to be physically active. So what they've shown is that they have increased appetitive value of running, enhanced frustration. If you put wheels in their cages and lock the wheel, you see a lot of frustration behavior. Increased hippocampal BDNF, and we're going to come back to BDNF because that is probably the one gene that's been shown over and over again in studies to be important in spontaneous activity. Increased FOS activity, that's just showing the brain as being very active, and increasing dopamine D2, D4. It is absolutely super intuitive that stimulation of reward systems during physical activity would be associated with the persistence of the behavior, right? Stimulation of those reward systems of any behavior is what drives things like addiction. So in this paper, this is again by Ted Garland and colleagues, basically what they showed was when they modeled the gene expression, they showed that it was distinct in different parts of the brain. So parts of the brain that are more focused on motor control, like cerebellum, might have a totally different pattern of gene expression than the striatum. So, but both of these parts of the brain have involvements or intercalations with the reward systems. In humans, when you do physical activity heritability studies, the estimates range pretty widely from about 10% to almost 85%, and that really depends on who you measure, right? Different populations, different types of cohorts, and certainly our ability to measure physical activity, spontaneous physical activity, is somewhat limited. And then just like in the mice, it's pretty clear that the genetics of physical activity and inactivity are probably driven by different kinds of genetic processes. So this is just a generic slide to show that when you're talking about diet, exercise, and weight loss is greatest with a combination of diet and exercise, and that's very intuitive as well, right? If you're impacting energy balance both at the energy in and energy out, you're probably going to see more weight loss. But weight regain is greatest when all you do is cut your diet. And both response and adherence to all kinds of interventions are highly variable. So how do genes influence exercise response? So this has been the work of my lab for the last about 15 years. And it took a mentor. I know there's trainees in the lab, in the audience. It took a mentor, Tony Jackson, who knew nothing about genes. He knew a lot about exercise. You know, whatever genes, Molly, sure, I'll help you do it. So he helped me put the study into place at the University of Houston. And basically the purpose of the Tiger study is to describe the genetic basis of response to exercise, to characterize individual variation, and to look at differences by gender and race. So the people in the study, there's been about 3,700 people, a little more than 3,700 people have gone through the exercise protocol. It's a 15-week protocol where they're highly regulated. They have the option to continue in this study past 15 weeks. They get an evaluation, a fairly basic evaluation of body composition and physical measures. They answer a whole host of questionnaires that are related to how and whether they'll respond to exercise. And of course, they give us some blood so that we can collect blood analytes and we can extract DNA. 3,665, so just under 3,700. So what we have for the study is every single person who works out, every time they work out, they wear a computerized heart rate monitor. So their exercise prescription is 30 minutes at 65 to 85% max heart rate, 3 days a week. When we wrote the grant in 2001, that was the recommendation of the American College of Sports Medicine. And those recommendations have gone up and down, up and down. You probably know that the recommendation is 150 minutes a week right now. But if you're exercising at high intensity, this recommendation is still consistent with the Institute of Medicine. So this is high intensity exercise, 65 to 85% heart rate reserve. It's not moderate to vigorous. Get your tennis shoes on and walk at lunchtime. So every time somebody worked out, we recorded their workout. We have more than 150,000 workouts recorded. And we have minute-to-minute recordings of their physical activity, as well as documenting the mode of exercise. So they were exercising. This half of the cohort, it was at University of Houston. Half of them were at University of Alabama at Birmingham. We have documented they're allowed to go onto the exercise floor and pick any kind of cardio piece of equipment that they want. So we've documented mode of exercise as well. And then we've used those data to calculate an empiric value of exercise dose. So basically, in this calculation, we adjust the time that they spent during exercise by the intensity that they exercised. So for example, if you exercise for 30 minutes at 50% intensity, that would be 15 intensity minutes. So for each exercise, their intensity, it ranges between 65 and 85%, although sometimes it goes higher, that is calculated, summed over all their workouts, and every person in the study has a score called the heart rate physical activity score that is an empiric measure of their total exercise dose. So I'm a big believer, and since I get a chance to talk about not just genetics but exercise, that one of the reasons why we have an inactivity problem is because people don't know how to exercise. And that sounds really silly sometimes, but in fact, everybody assumes that you know how to go out and exercise within your heart rate zone, and you know what that feels like. And in fact, what most people do when they say, I'm going to get in shape, they go and they work out crazy hard, and they get really sore, and exercise was a terrible experience for them, and they don't ever want to do it again. And I'll show you the heart rate data to support that idea, because here's the zone, those two green lines, and this is time, and this is heart rate. So this is a real person's workout, and I've heard this so many times from the participants in the study, where they say, I'm sorry, Dr. Bray, I'm going to have to quit. This is just too hard. I got in over my head. And what I say to them is, you worked out really hard. We immediately look at their heart rate monitor. You really went crazy. You're working out way too hard. Just slow it down a little bit. So here, this person, they slowed it down, and what happened is they said, I can't do this. And I don't know if you can see this, but their workout their heart rate monitor accumulates intensity time in the zone whenever they're in the zone and doesn't accumulate any time even though they're still working out. So this workout was almost 70 minutes long. And this person said, this sucks. The second time, this sucks. I can't take 70 minutes to get my 30 minutes in the zone. And we looked at the data and said, you're bouncing around just below your zone. So take your headphones off and listen to your heart rate monitor, which beeps at you when you're working out too high or too low, and see if you can get in the zone. And so here they are. This is the third workout. This is real data, bouncing around in the zone. And when they get here and they hear beep, 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 they slow back down. And the great thing about when you're not fit, it doesn't help you when you're fit, but when you're not fit and you go below the zone, you start to walk, your heart rate still stays pretty high. You can be walking and you feel like you're chilling out, but because you're less fit, your heart rate is still in the zone, and it takes you to walking a pretty long time for you to get out of it. So these are real data. Now she's saying, I'm listening to my heart rate, I can do it. This is getting really easy. And the funny thing is this is getting much easier, and that person was at the top of their zone almost the whole time, which is a pretty hard workout. 85% heart rate reserve is really hard. And then when they finally figure it out, what you can see is they never hear their heart rate monitor. They get just within the zone, they stay just within the zone, and now we've taught them with a little bit of feedback how to exercise at an optimal place for them. And that's the most valuable thing probably that we do in the Tiger study. So I feel really proud of that because we see those kind of data all the time. So here's just a kind of a distribution of the data. You can see at University of Houston, and Houston is it was the second or third majority minority city, so it's a lot of everybody, not quite as spread out at University of Alabama at Birmingham, but very similar data between the two cohorts. And what you can see, and this is not really surprising, Tiger study is not a weight loss study. If you do the math, there's no way that 30 minutes a day, 3 days a week is going to induce a lot of weight loss. We do see significant weight loss, and we know absolutely that it has to do with adjusting their diet. But we see these relationships that we expect to be true, that those people who lost the most weight worked out at the highest intensities and for the longest duration. So that's a little bit of basic energy balance mathematics. But what we also know, and I didn't include those slides, but I should have, is we have gone back and looked at the relationship between intensity and dietary intake. We see that those people working out at the highest intensities eat I would say the optimal, these in terms of dietary patterns, they eat the optimal healthy dietary pattern, and that going from sedentary, which all of them are at baseline, to physically active, those people who worked out through that at the highest zones are more likely to change their diet from unhealthy to healthy, which is pretty cool. So we know that diet has a lot to do with this. It's not just at physical activity. And again, those people who are adherent to the exercise protocol. And now this is calculations on the back end. So I will tell you that when you're in they take a class for college credit. So not only do we not pay them, they pay us because they have to pay tuition to be in the study. And so which turns out to be a really serendipitous, but I totally attest to that it's serendipity to have thought of the study design, because it gives them a framework in which their likelihood of adhering is even higher because they have a goal besides just seeing me every day and being embarrassed to see me when they drop out of the study, right? So they want to get their A. And I always tell them it's the easiest A they'll ever make, but yet people do drop out and some people are non-adherent. When they come to class, they're told if they didn't get their heart rate in their 30 minutes in their zone, they're told you didn't get your 30 minutes, your workout technically doesn't count. That's just to get them back on track. It sounds horrible saying that to you. But in fact, they get all the credit when we calculate their total exercise dose. So this is total exercise dose, adherent or non-adherent. And you can see that if you adhere to an exercise program, obviously you're going to see more positive change. So this is the variability in exercise response. And you guys have probably seen these these waterfall plots for many different kinds of, of data. And this is to show you the variability, and this is to show you the amount of residuals accounted for by things like gender, demographic variables, and exercise parameters. So you can see that there's still a lot of variability in exercise response unaccounted for by things that you think would explain variability in exercise response. That's in terms of BMI, and this is in terms of body weight. So at least some of that we think is accounted for by genes. So my collaborators, this is Anna Vasquez, has been my collaborator for many years, and Fei Dong was my postdoc at the time. This is just showing you the race ethnicity distribution of the tiger study based on their genotypes across this chip, the metabo chip. And this is calculated based on genotypes, calculated heritability of both baseline measures of body size as well as change in measures of body size. So all of those estimates are significant. The cohort is fairly large, so that explains some of that. The strongest heritabilities for females are for fat distribution. So you see that that hips and waist-hip ratio and waist are highly heritable at baseline. So basically cross-sectional data. And then in terms of males, change or response to exercise, the heritability of responding is actually higher for almost all traits in males compared to females, with the exception of waist-to-hip ratio. So waist-to-hip ratio, highly heritable, changing your shape in terms of the ratio of your waist to your hip. So those are heritability estimates from the genetic data. These are genomic correlations. So these are looking to see how much pleiotropy or how much cross-correlation there is between these traits. This is cross-correlation at baseline in the bottom triangle, and this is cross-correlation between traits and the top, for change in the top triangle. So in terms of females, females show stronger genetic correlations at baseline in the cross-sectional data, and males seem to respond and have more cross-correlations in terms of change. So suggesting that some of the same genes may influence change in more than one anthropometric trait. These are all just anthropometrics for males. And so this is the whole genome analysis. I'm not showing you the SNPs, but I'm showing you the pathways. So we've done the whole genome analysis of body composition response, and this is response, this is change following exercise. So in terms of the genes that were significantly associated with change, in females, the two pathways that are most prominently associated with change are the Cori cycle, with, if you remember your metabolism, the Cori cycle is one way your body clears lactate and recycles lactate so that you can use it again. So that one actually makes a whole lot of sense. And then the FTO, because we're talking about body composition and adiposity, FTO also makes sense as well. So these are the prime genes in those pathways. In terms of males, the pathways, multiple hits on lipid metabolism pathways. So in terms of body composition, it looks like, at least for these data, that lipid metabolism seems to be a big player. And then when you combine the samples and control for gender, you see that FTO comes up again. So how do genes influence exercise adherence? So that's response, and that's response in terms of body composition. I'm just showing you body composition today, but what about adherence? So one thing that everybody, I think, would acknowledge is that I got in trouble at the American Society of Nutrition by giving a talk and starting it out by saying, all diets work. It doesn't matter if you eat protein or carbs or fat or whatever. And you're nodding, Jack, and I know that Kevin would support that. But people who dig in and say, it's carbs, it's protein, it's fat. In fact, all diets work. And it's not the diet, or it's not even the exercise program. It's not the great tiger study, although, of course, the tiger study is the best exercise intervention ever invented. It's getting people to stick to it, right? And so there's a whole long, very deep research base of people looking at what influences whether or not people change. And all of the things listed here have been suggested or reported to be strong influencers of whether or not people stick to an exercise program. In our study, age, not a predictor. We don't have a very big distribution of age, so that's not surprising. Gender did not differentiate anybody. Baseline activity did not influence anything, any kind of adherence. Self-motivation. So I often get asked, what? You just didn't ask the right psychological questions. In our data, we have pretty extensive battery of psych questions, and self-motivation was not predictive. But BMI, waist, and genes. So here is the whole genome association for exercise adherence. So in this case, it's adherence in terms of exercise dose. So that calculated empiric value of how much exercise did you get. These are the top hits, and I'm just going to focus on two of them for today's talk. BDNF, as I mentioned it earlier, it is probably the one gene that's been shown over and over again in both animal and human studies to be associated with physical activity and other kinds of physical activity traits. And FTO, which probably many of you are familiar with FTO, it's probably the most strongly replicated obesity gene. Did you have a question? BDNF, well, just under, yeah, it looks like it in this case, but so actually that's a little bit misleading because this chip, after you get rid of the monomorphic, after you get rid of all the monomorphic variants on this chip, then the adjustment for FDR adjustment is 10 to the minus 4.8 or something like that. So BDNF is right on the cusp. Those lines of significance are kind of auto-generated, but I'm glad you pointed that out because I should fix that. But it did just make the cut. No, because it's not a super dense chip, and once you get rid of the monomorphic, polymorphisms, there's probably about maybe 150K. So that particular chip, the metabo chip, it's a semi-mapping chip, but it's really a candidate gene chip. So it's semi-mapping because it does cover the whole genome, but it's really densely populated within candidate genes for obesity and cardiometabolic disease. So it's not truly mapping the entire genome. Yeah. Yeah, exactly. So this is just some data from other groups to show that. So FTO, it pops up here in our study as pretty strongly significantly related to exercise adherence, whether or not people stick to or can adhere to their exercise prescription. And this data from the Helena study, and then these are just some more examples to show that FTO, in fact, has been shown the risk-raising effect of FTO in increasing risk for obesity has been shown multiple times in several different populations to be attenuated by physical activity. So if you're physically active, that risk-raising effect of the FTO risk-raising alleles seems to go away. So this is old data from our group. We're finally going to publish it. This is some original data where we were looking at 13 tag SNPs for FTO and basically showed that 5 of the 13 raised risk for exercise dropout. And when you combine them, any 3 alleles more than doubled your risk for dropping out within the FTO gene. This is the original gene that was identified as being associated with obesity, but FTO is a really big gene, and even now people are still trying to figure out, that was 2008. People are still trying to figure out what FTO actually does, although we have some more clues. So all we did was we pulled out the Tiger participants based on whether or not they had less than 2 risk-raising alleles or more than 6 and compared them. And in fact, just pulling them out by their risk-raising genotype at those 5 loci showed that the low-risk group exercised longer and the low-risk group had greater exercise tolerance. So kind of a validation that the odds ratios that we generated are supported by quantitative data. So here, these are some structural equation modeling of the effects of the FTO controlling for the psychological measures. So this is the work of Rod Dishman, who's a very, very famous sports psychologist, basically controlling for measures that we had collected based on self-efficacy related to physical activity, self-motivation, enjoyment, processes of change, behavioral and experiential. So basically modeling the effect of the FTO variance, controlling for the mediating effects, mediating might not be the right word, effects of psychological measures known to influence whether or not you adhere to exercise. And in fact, what Rod's analyses have shown is that there are some FTO variants that seem to act through psychological measures, either influencing the psychological measures or combining with psychological measures to influence whether or not a person is compliant. And there are some FTO variants that seem to act directly on influencing, compliance independent of all of the psychological measures. And all of these analyses are already adjusted for gender, age, percent fat, and your baseline physical activity level. So pretty interesting and a different kind of statistical approach that supports these relationships. When we look at whole genome gene expression, so in a very small subset of the sample, they were again stratified by whether or not they had less than 2 risk alleles or greater than 6 risk alleles. This is the only gene that was significant. Now, we're redoing all these analyses because of the way the way the software that we were using was limiting us from doing two-way analyses. But this was the only significant gene, DGKC. DGKC is diacylglycerol kinase zeta. So this was the work of an honors student. An honors student was working on this part of the data. She said, oh, diacylglycerol. Oh, it has to have something to do with lipid metabolism. She was focusing very much on the lipid metabolism aspect. But in fact, diacylglycerol is a really interesting signaling molecule. And it's been associated with several things besides lipid metabolism. So this is the kinase zeta, the zeta version of this kinase. It's associated with increased muscle mass. It's associated with immune function and with cancer. And interestingly, it's associated with heroin, potentially with heroin and cocaine addiction, cerebellar depression, and higher brain function. So we don't have any gene expression data in brains of people who participated in the Tiger study, but the biology is very interesting in that this molecule would fit a hypothesis that things are happening in the brain that are influencing why people are persisting in exercise. So in terms of FTO, it's highly expressed in the brain and in the pancreas. Its expression increases. When it was discovered, people thought, oh, it's going to be another satiety factor because all the obesity genes till that point were all satiety factor genes. And it is upregulated after food deprivation, and it's associated with satiety regulation, but has not been shown to be a clear satiety regulating factor. It has been shown to act as a demethylase and a deoxygenase, and how that influences its downstream targets is not super clear. We took a subset of the Tiger sample, again, pulled them out, stratified by whether or not they had less than 2 or greater than 6 FTO risk-raising alleles, and we ran these methylation arrays on them. So there were 739 genes identified, 574 after adjusting for multiple testing, and of the genes that were significant, the top gene is the STAT3 gene. So just to show you a little bit closer up, this is 2 risk-raising alleles in blacks and whites. This is 6 in blacks and whites. And this is pre-post. So you can see not much of a relationship here, a little bit maybe right here within this group, but very clear demethylation from pre to post exercise for these people with more than 6 risk-raising alleles in FTO. The STAT gene was demethylated from pre to post exercise, and demethylation was significantly associated with FTO genotype. And just to remind you that DGKZ is our top gene expression hit in this small sample. And in fact, one of the binding sites, one of the transcription factor binding sites for DGKZ is STAT3. So linking our top hit in gene expression to our top hit in methylation. So I'm going to cover a little bit of brain-derived neurotropic factor and then circle back. So we know that BDNF is really, really important for neuroplasticity. It's involved in lots of processes in the brain. It is one of the most consistently associated. It was probably the first gene identified as a potential physical activity gene in animals. And it's also associated with a range of mental states, cognition, depression, schizophrenia. And we also know that exercise reduces depressive states and also has been shown to reduce BDNF expression. So in the Tiger study, a subset of the participants were given a visual analog scale to complete pre and post exercise. So they come to the checkout station to get their heart rate monitor, and they're asked to, this is a little touch screen. It was a cool story because a little undergrad, we were having a lab meeting, and I said, I wish I had something, because we were doing all this by hand, measuring literally visual analog scales with rulers. All of you have done that before. You know it's a painful process. I said, I wish I just had a little touch screen that somebody could touch. And the very next day, he came up with his phone and said, is this what you had in mind? He had written the program. So he wrote the program. This was not a phone, but a bigger screen. And basically what the participants do without thinking is tell us, how do you feel prior to exercise? And then they do the same exact thing after their workout. And what we're interested in is not so much whether they're depressed or elated or whatever, but whether or not they're labile to changing as a consequence of exercise. And so these are the data to show you the prediction of exercise dropout from mood. And this is change in mood state from pre to post. So change positive or negative. So this is changing from a positive state to a negative state is significantly predictive of exercise dropout. The change in tension and change in fatigue. So the anchors for tension are tense and calm. The anchors for fatigue are tired and rested. Tired and rested or something like that. Tired and energetic. We had elated. I have to laugh because we had elated on there, and there were too many college-age kids that we had to explain what elated meant, which is really sad. So we had to change it. So kind of interesting data to show that how your mood changes affects whether or not you persist in exercise. And again, it's very intuitive. It makes sense. If my mood is changing in the wrong direction, if I'm feeling more tense, if I'm feeling more tired and uncomfortably tired, that may lead to the fact that I'm going to drop out. So these two are significant. So this is kind of this is an analysis of, we said, well, would those responses to exercise be the same for people who just began to exercise versus the ones who had exercised for a while? So I showed you the earlier data that showed within about 4 or 5 workouts, you acquire enough fitness. It was purposeful to show you 5 sets of data in the heart rate monitor because I always say, give me 5 workouts. By the time you've worked out 5 times, you got the hang of it, but you also have acquired enough fitness so that some of the initial discomfort of exercising is gone too. It doesn't seem like in 5 workouts you're going to be terribly fit, but if you're going from sedentary to active, those 5 workouts make a difference. So this is looking at change pre to post in those values of tension, hunger, fatigue, and depression within the first 2 weeks of the study and within the last 13 weeks of the study. So what you can see is that people do feel more hungry. So this is the green is hunger, and the yellow is tension. Negative is a decrease in those feelings, and then positive is an increase in those feelings. And so interestingly, people tended to feel more tense after their workout, which is a kind of counterintuitive. But as they start to become more acclimated to the workout, they feel less hungry after the workout. And so these are controlled for time since previous meal. In terms of BDNF, we also see that there's a single genotype. We didn't genotype the full array for these guys, but we see that there's a single genotype in BDNF that is strongly associated with whether or not they change their depression score pre and post exercise. So there's some interesting biology that links some of our findings. Ireson is a molecule that was discovered not that long ago by Bruce Spiegelman's group, but basically arises when you exercise, rises from skeletal muscle, and it's been shown to stimulate STAT3 signaling and ultimately stimulate hippocampal gene expression, and in turn, both directly and indirectly increase BDNF. So this particular paper is a little bit about Alzheimer's disease, and one of the mechanisms by which exercise, which has been shown pretty strongly to attenuate Alzheimer's symptomology. And so this is a kind of just an interesting biology that links some of our major findings. In terms of FTO, again, we still see we see some associations with FTO variants. So this is back to the original genotyping panel. We see that FTO variants, these are 0, 1, or 2 copies of the minor allele, significantly associated with tension, multiple variants associated with change in fatigue, and a variant associated with change in hunger. So when do health behaviors become unhealthy? One of the ideas behind these pictures is to show that there was probably a continuum of behavior. There's not adherent and non-adherent. And so looking at yes, no variables that depict adherence, maybe is not the way to go, and that there's this continuum. And I wasn't exactly sure where. I started to put adherence in the middle, but I would say for anybody who studies adherence of anything, then we had a meeting, and actually it was arranged by NHGRI folks, and maybe about, I don't know, five or so years ago, where we were all in a room, those of us studying all kinds of different kinds of adherence. And the purpose of the meeting was to sort of brainstorm, are the same genes influencing perhaps this distribution of behavior from non-adherent to addicted, and whether or not adherence and compulsion are properly placed on the distribution, or whether or not the distribution is normal. Please don't, you mathematicians, statisticians, don't ding me on my little figure here. So the question I'm asked all the time is, cool, you're looking for genes, that's terrific. But what are we going to do with it, right? And so this was, for those of you who are old like me, you know that this was your sequencer. You were the sequencer, basically. And you took the gel with all the little bands on it, and you put a little ruler up to it, and you literally by hand called, I'm looking at the young people, you have to appreciate this. You literally called bases, T, A, G, C. You read them from your gel. And of course, we know that now we've got amazing sequencers that can sequence a whole genome in less than a week or a little over a week. And this is the sequencer that will probably, something like this will enable us to use this kind of data in the field. This is called the Minion, which the company was kind of upset when they realized they had named it Minion. But it is a sequencer. It's not a whole genome sequencer, but it can sequence quite a lot of sequence. It plugs into a PC or laptop. You can put whole blood on it or whole saliva on it. It takes blood, serum, urine, and you can generate fairly long reads from this. So in the clinic, and it has a USB interface, so it does its thing, you plug it in, and you get your sequence. In the clinic, it's very intuitive. It makes a lot of sense if I'm running a cystic fibrosis clinic. I can sequence your cystic fibrosis gene, possibly multiple genes, and tell you and have some decision making about my prescription for your treatment. But this little guy costs $5K. That's a doable thing to have in places where maybe you have someone, maybe a weight loss clinic, maybe you have someone who feels resistant to exercise. This is something that can guide you. And it's not so much everybody, and I also hear many, many times, millions of times, everybody should exercise. well, you're not telling us anything new, Molly. But in fact, some people have been shown to have deleterious consequences of exercise. And it's not really the average person who might respond to exercise. It's the person who, like many people in the Tiger study, doesn't know they have the potential to be highly responsive. And it's certainly the person to identify early who may be the early dropout who needs that extra support. And we know that right off the bat, and we help them with their compliance. So just to conclude, genes associated with exercise response in terms of body composition are primarily associated with metabolism, the Cori cycle, the obesity signaling, and lipid metabolism. Genetic effects of exercise adherence seem to have neural underpinnings both in animals and humans, and multi-levels of omics data are designed to reveal maybe new pathways. So I showed you just a tiny little bit of our data, but we just got some funding to add metabolomics to the Tiger data and to extend the gene expression data, and that will, we hope, reveal a whole host of pathways, or at least more strongly identify the ones that we've already got data to look at. And the technology exists today to use these data in the field. So I just want to thank all the collaborators, all the many students, grad students, postdocs who worked on this project. And I'll stop there. I do have a little bit, I want to leave some time for questions, but I have a little bit of circadian data if anybody wants to see it. But I'll stop there. So thank you very much. Yes, sir.
Thank you very much for this great lecture. I think that your force of integrating the key question is when will people respond positively to exercise? And the only behaviors you can train are those that are rewarded. And I think that that's a key philosophical question that you're pursuing. So I was surprised that your gene chip that you used was a metabolomic gene chip, looking at lipid and calorie metabolism. I'm wondering if you have thought about, or if you have data available to look at addiction gene chips and reward system gene chips in your same population, which may uncover, may go to the heart of reward physiology and might give you more information about a new hypothesis.
Yeah, well, what I would really love is a proper mapping chip so that I can look at the whole genome. That would be great. So we happen to do this. The original study was funded to do a small panel of candidate genes, and of course times changed, and we maximize that money by using a chip. But really, what we need to do is a whole genome mapping chip, and then that yes, we do. Do you have any? Yeah. Yeah, they actually have quite a lot. Yeah.
So I have two questions. One about the end, which is more philosophical, and one is more technical. Leave it to you.
Oh, you decide.
Well, we did philosophical, so we'll stick with that. The trivial fraction that you're finding for people who drop out is pretty small. And does that bring up issues of, presumably the attributable fraction of the genetics for people who succeed is also. And so if you're going to use that for motivation and personalization, as I understand it, is a really good motivator.
Well, you know, I'm glad you asked that question, because the data don't show that genetic information are generally motivating, even in people who are not just at risk for dropping out of exercise, but maybe more serious kinds of things. But I wasn't presenting it so much as a means of motivating people to exercise as a means of informing my decisions about how I intervene on specific populations, right? So if I know, if I have strong evidence that a collection of genotypes puts people at risk for dropping out, and I know who those people are, then I can focus my efforts on those guys, because I can tell you that the people who discovered, oh wow, I didn't show any of the, I have some wonderful photographs of people who are just changed their whole life, had no idea that they could be so responsive. Those guys, all you need to get them is to get up off the couch, right? But it's the people who come, and even in our study, I will totally admit that there were people in the study who did not respond to exercise and were very, very frustrated and would say to us, well, I came here, you're supposed to be smart, why didn't you help me? And that's really frustrating. So I would say, from a practitioner's point of view, if we're going to consider exercise a prescription, we need as much information as anybody prescribing any kind of treatment would need to optimize that treatment. So that's my philosophy.
Now, the technical question for the expression data, have you looked into GTEx? I don't know how common the alleles you were looking at are, but in GTEx you do get the brain and you get all the rest of the and you get the phenotypes.
Yeah. So we haven't compared directly with GTEx. That's a really small sample. So that sample was really small, and it was also analyzed. The analyst analyzed it two-way ANOVAs and not 2x2s, and so not taking into account both timing and group. So I hesitated to show it, but since it's one very strong effect, if anything, it's conservative. We'll see something else. But so yeah, there's other things that we need to do. And also, to your point about technicality, the reason why we haven't published this quite yet is we were all ready to go, and we decided that this chip is fairly old and we needed to remap it. And as it turns out, the mapping is significantly different for probably about a third of the chip. So we're maybe not quite that much, but a good bit of the chip is either. The other thing that we discovered is that as Illumina annotated the chip, if the SNP was intergenic, they just annotated it with the 2 genes closest, some of them which might be very far away. And so if you put all of that in your pathway analysis, then you have a lot of noise and crap that's not helping you. So anyway, we're redoing that. I showed you the unadjusted. I hope I don't get dinged on that. But we're redoing all the chip mapping and kind of redo the analyses just to make sure that what we're reporting is correct. So I can tell you something sort of interesting that we're doing. We have this funny study. So we're interested in circadian rhythms as well, but we're also interested in how exercise may influence circadian rhythms. So just share this with you. This is hot off the presses. We were interested in shift work and how shift work influences disease risk. So this is a model in which the shift rotates every 3 or 4 days, just like it does for nurses, 3 on, 4 off, 4 on, 3 off. And so the animals get a changing light all the time. And we had this hypothesis that if you give them a bout of exercise, one bout every 24 hours, whether they're on shift or off shift, that it might help maintain skeletal muscle rhythms. And the idea is that if you can maintain skeletal muscle rhythms, you might be able to maintain insulin sensitivity, and that would be really important. So that was our hypothesis. And then we said, well, maybe if we marry eating with light, with activity, then that will also influence circadian rhythms. And so if you were on shift and you only ate on shift, maybe you'd be healthier than if you ate all throughout the day. So those were our 2 hypotheses. And so this is just the study designed to show you light, dark light. That's what the things are. And this is just to show you the statistical comparisons. All of the animals are in this feeding system where we can control their access to food and marry that to light. This is their little running wheel. Oh, that got messed up. So a Bluetooth running wheel that we can remotely access their activity patterns. This is the control group that sees regular light, dark all the time. And this is the shift group. So basically, sort of patterning in the very beginning. This is the first week that they're exposed. This is on high and low fat diets, but quickly losing rhythmic patterns. And we were completely wrong. This study is halfway through, and I don't know why I want to show it to you. It's just so interesting to me. We were completely wrong. These are glucose tolerance tests. And the people the mice that responded the worst by far the ones that we messed with their eating patterns. So the groups that had meals paired with whenever lights were off, our hypothesis was that that would mimic a nurse who only ate when she worked, or a shift worker who only ate when she worked, absolutely had the very worst glucose tolerance. So these two groups up here are groups with and without a bout of exercise. So absolutely not consistent with our hypothesis. And this group down here is the animals who have ad libitum access to exercise wheels in the face of changing light-dark cycles, which we know their brains are completely screwed up. All you have to do is expose animals to about a week, maybe even a few days of altered light-dark, and you already get your brain rhythms really messed up. But this ad libitum access to exercise, wow, what a powerful argument that you should exercise a lot if you're a shift worker. This is on high fat and that's on low fat. You see the same, kind of the similar patterns. Not quite the same on low fat because the animals don't exercise as much on low fat, interestingly. And this is just area under the curve. But anyway, we just got those data. I was like, that's so cool. I'm going to show that at NIH. But that's it. Thank you so much.